All right, so this is basically what we talked about last time, specifically the details of the MHC Class I presentation and processing pathway. And if you remember, we divided this process into 6 steps. Those 6 steps were the acquisition of antigen, tagging of that antigen for destruction, proteolysis, delivery, binding, and display. And in the case of MHC Class I, acquisition happened sort of passively by the process of infection or having a protein in the cytoplasm. We tagged the proteins to be destroyed by adding ubiquitin. We did proteolysis in the proteasome. We used TAP to deliver the peptides to the class I. The chaperone tapicin allowed binding to happen, and the display part of the process was constitutive. So that was what we talked about on Wednesday. And that is, again, all summarized on this slide. Also, kind of just remember that this is being done by all nucleated cells. And it's a constitutive process, so this is basically being done all the time by all of your nucleated cells. Yes, under certain conditions, you can increase the amounts on the surface. But right now, you are processing and presenting MHC Class I on the surface of all of your cells. In addition to the fact that you're doing VDJ recombination, you're quite busy immunologically. And remember last time that we kind of talked through details of the biosynthetic secretory pathway. By and large, the MHC Class I presentation pathway that we talked about last time is restricted to proteins from this sort of light blue portion of the cell. So remember, I kind of told you that immunologists like to divide the cell into the secretory pathway, or the yellow, and the not secretory pathway, or the blue, the cytoplasm. So Class I was really about presenting peptides from this light blue area. Today, we're going to have the other side of this, which is the MHC Class II presentation and processing pathway. MHC Class II addresses the other portion of the cell, which is the yellow areas here, proteins that are inside the biosynthetic secretory pathway. And so that's a big part of the distinction, is where that protein was in the first place. So here you can see the MHC Class II information in overview. So what you want to remember about MHC Class II is that it is made up of 2 chains, an alpha and a beta chain, that are about the same size. If you actually look back to the genome thing, I didn't push this in the genome thing because it's not a big deal for most of what we're talking about. But you actually did see like DRA, DRB. Like there are actually 2 genes for each of the Class IIs. It's because there's the 2 chains. And those are encoding those 2 chains. This is folded correctly. Hello.
Hi, is this your immunology class?
It is.
Okay, I found an iPod case that says immunology on it in the back room. And I thought this would be a good candidate, if possible. Okay. Is that yours?
It's not mine.
None of yours? So okay, I'll take it to the dean's office. If you know of anybody, I'm sorry for interrupting.
It's okay.
I just figured that this.
These do seem to be the right suspects for that.
There are pods in it, but anyway, I'll take it to the dean's office. Okay,
Okay, no problem. No worries. So we don't really have the class 2 folded correctly unless it has a peptide in its cleft, just like with class 1. And remember that class 2 plus peptide is an incredibly stable molecule. It is known biochemically as being SDS stable. So SDS is a typical way that we denature proteins, and it does not denature this complex. So this is a very, very stable complex that is going to come up later. MHC Class II presents peptides to the T cell receptor, specifically on CD4 T cells, because we have this protein CD4 that is also interacting with MHC Class II. And MHC Class II presentation is really done by 3 types of cells in your body, B cells, macrophages, and dendritic cells. This also says T cells. That actually depends on the species, so you can ignore that. It's really just B cells, macrophages, dendritic cells. Sometimes some cells in the thymus are sort of the exception. And as we will see a little bit today, and even more so a little bit later, this process is regulated. So these cells are not presenting on class 2 all the time. They're only presenting in class 2 kind of when we turn on that process. Note that these cells also all have class 1. So they present on class 1 and class 2. They're doing both of these things at the same time. So this is the lovely figure from your textbook that goes through this whole process. And as I already mentioned on the previous slide, class 2 differs from class 1 in that it is presenting peptides that are in the endocytic pathway. So they're in, or the biosynthetic circulatory pathway. They're already in this process. They're not peptides from the cytoplasm. So that's kind of a big difference here. We can think about the same 6 steps for MHC Class II presentation that we thought about for MHC Class I presentation. And so we're going to go through the same set of 6 steps and see how they work for Class II compared to Class I. So for acquisition of antigen for MHC Class II, the sort of most common way we can think about acquiring antigen here is phagocytosis. So this is very different than what we saw in Class I. Because in this situation, the cell actively has to do something. Whereas in Class I, the cell sort of by default had the thing in its cytoplasm. Here, the cell actually has to try to acquire the antigen. And that will happen by phagocytosis. If you remember the slide I just showed you, 2 of the types of cells that present on MHC Class II are really good at phagocytosis, dendritic cells and macrophages. And so this kind of ties that together. This is sort of some general cell bio information about different ways materials can be internalized in a cell. So you can see here some aspects of pinocytosis. These are actually the clathrin and the other aspects of the vesicles and the calveoli can all be kind of thought of as being types of endocytosis. And then you can see phagocytosis here. And the reason why I'm showing you this is you can see that phagocytosis is super different than the other ones in terms of the size of things that are being internalized, and also in terms of the amount of work the cell has to do this process. The cell actually has to kind of completely rearrange its cytoskeleton and its membrane to bring in something this big compared to these little things. So phagocytosis is not like a small deal for the cell. That's why only some cells can do it, because it's actually like a whole big process. And so you might, so remember, actually, you can see sort of the details of what happens when something is phagocytosed here. We've got this big reorganization of the membrane as well as the cytoskeleton to put those materials into a new organelle. Since that organelle was generated by phagocytosis, we call it the phagosome. But that phagosome will eventually kind of merge with endosomes and the lysosome, and so it ends up in this pathway that we've been talking about. But what you also might realize is that I told you that there are 3 different types of cells that are able to do MHC Class II presentation, which are dendritic cells and macrophages, but also B cells. We've already heard a little bit about B cells and the way that B cells can function, and we didn't ever hear about phagocytosis, because B cells actually can't do phagocytosis. And so we might think about, well, then how the heck do they acquire antigen? And there's one other way that we can acquire antigen, which is really the big thing that we do in B cells. And really that has to do with endocytosis. And again, you can see that it's pretty similar to phagocytosis. We're just bringing things into tinier organelles. And when we make them, just like we do phagocytosis and we make a phagosome, we do endocytosis and make an endosome. B cells will bring in something that will end up on MHC Class II via a process that's known as receptor mediated endocytosis. And so what happens here is that our B cell receptor, there's receptor in the name of this process, our B cell receptor binds to antigen. And in fact, when the B cell receptor binds to antigen, one of the things that happens is the B cell actually endocytoses the receptor plus antigen. It will actually separate due to acidity the B cell receptor from the antigen. The antigen will go right back up on the surface, or the B cell receptor, the antigen will stay inside to become peptides. And so the B cell can use its B cell receptor to bring in an antigen to get it ready for Class II presentation. And so this is how B cells will acquire their antigen for this process. You can see some details of this process here as well. And so again, I'm going to take just a quick kind of point about some aspects of general cell biology of the biosynthetic secretory pathway. So when we are, so all of these compartments, the lysosome, the endosome, the phagosome, et cetera, they're all actually interconnected with one another. When we first do endocytosis, really we usually call something that we make an early endosome. Or if it just comes off of the Golgi, it's an early endosome. But we can actually have that vesicle, that compartment, change in different ways. Sometimes it changes by changing. Sometimes it changes by fusing with other vesicles and sort of gaining their stuff. I'm not going to really distinguish between those two options here. But we can get things like late endosomes, lysosomes, things like that. So these are all just sort of interconnected types of organelles. One big difference with a lot of these organelles is that when you get to the lysosome is very acidic. And the pH sort of decreases through this process. So here, you're maybe a teensy bit acidic. And here you're a bit more acidic, and here you're a bit more acidic, and here you're super acidic. So one thing that happens along this process is we get more and more acidic. And whether that's because the thing changes itself or fuses with somebody else, don't worry about. And so you can kind of see sort of the progress of this pathway. There's one other, and so what this actually can lead to, and why this is used in the cell normally, is that this very acidic lysosome is a place where we destroy things. The lysosome is full of enzymes that are used for destruction. It's full of lipases to destroy lipids, proteases to destroy proteins, different glycosidases to get rid of carbohydrates, all of those things. In a lot of ways, the lysosome is the trash can for the secretory pathway. Just like the proteasome was the trash can for the cytoplasm. So that's kind of just generally, if you ever want to degrade something in this pathway, it goes to the lysosome. There's one other compartment that is a part of the biosynthetic secretory pathway that I actually learned about in immunology before I ever learned about it in a cell bio class, so I just want to mention it here. It is called a multivesicular body. It is sort of in that continuum of different endosomes. It's usually kind of around sort of where the late endosome / lysosome is. Somewhere near the end of that process, we can have multivesicular bodies. And what happens with a multivesicular body is sort of like an endosome starts to try to like phagocytose itself almost. So here you can see that the multivesicular body gets little areas inside of it that pinch off. They make vesicles inside the thing, multiple vesicles inside it, multivesicular body. One of the reasons why this is really important is now things that were previously transmembrane proteins get put inside of the vesicle so that they can be degraded, so that we can destroy them. So this is pretty nice because now we can destroy these proteins because they're inside the vesicle. So if we put destruction enzymes here, we can destroy them. And you can see these are actually multivesicular bodies under the microscope. You can actually see all the like vesicles within vesicles in them. And so the multivesicular body is going to be a place where we're going to see some events happen later. So this kind of gets us to some of the next steps of this process. We acquire antigen by endocytosis or phagocytosis, and then we make an endosome or a phagosome. And kind of by default, like the way endosomes and phagosomes work, is that they fuse with lysosomes, or they grow up to become lysosomes. So almost automatically, when we bring in an antigen this way, it's ready to be proteolyzed. We don't actually have to tag it. If anything, tagging in this case is like being in endosome phagosome. And we do the proteolysis in the lysosome. Be careful here. Earlier in the semester, we learned about an enzyme called lysozyme. Lysozyme and the lysosome are not the same word. So the degradation's pretty easy. The lysosome is super acidic, and it's full of all sorts of enzymes. So once we've done phagocytosis or endocytosis, our stuff is headed to the endosome and to the lysosome. That's just how this pathway works. It's kind of by default going to end up in the lysosome. And the lysosome is going to have these enzymes. These enzymes are only active in acid conditions, which is good. That way, if they like get into your cytoplasm by mistake, they're not going to kill the rest of the cell. They're only active when they're here. This is sort of the trash can for the biosynthetic secretory pathway. So once the cell actually does the active thing of phagocytosis or endocytosis, it really doesn't have to do much active anymore. for tagging or proteolysis. And things, again, start to kind of get a little bit interesting when we get to delivery. So if you remember, in the case of the class 1 processing pathway, delivery was done by TAP. Delivery is sort of a key step for class 1. And this is sort of a question of delivery of that peptide to the empty MHC. We can also think a little bit about delivery for the class 2 presentation pathway. So this is a figure that is sort of adapted from one from your textbook. And you can see the biosynthesis of MHC Class II. It's biosynthesized in the ER because it's a transmembrane protein. We want to be presenting it on the surface of the cell, just like we saw for Class 1. So there's the biosynthesis of Class 2. This is kind of the route that Class 2 or any transmembrane protein would take to get to the surface of the cell. And this is the route that our peptides are taking to come in and get degraded. So if you look at this, what are your thoughts about delivery here, specifically delivery of the peptide to the MHC? Yeah, Jamie.
Directionality, like with the direction that the antigen, the peptides are moving, and with the direction that the MHC class 2 is moving, they're going to overlap at some point.
Yeah, it's like, well, this looks like a really easy one, because their paths overlap. They're both in the same place. So this should be super easy. And so if we think about delivery of peptide to MHC, it's easy. It's just be in the secretory pathway. But there's actually a problem here. And some figures from your textbook completely gloss over the problem. And I'm like, this is such a lovely figure, except why did you do this? It's sort of shown here. It's shown better in this figure from your textbook. Again, I have very slightly adapted this figure to get at the issue well. So here you can see the biosynthesis of both class 1 and class 2. They're both being biosynthesized in the ER. And then moving to the cell surface. And you can see that class 1 meets up with its peptides in the ER. Class 2 is going to meet up with its peptides on the way to getting presented. Yeah? Make sense? So what's the problem here? There is a massive problem here. Massive problem. And sometimes the way they draw this is like, tra-la-la-la-la, there's no problem. It's a huge, humongous problem. Yeah, Jamie?
Is it that MHC class 1 and class 2 are both being produced in the ER at the same time?
They are both being produced in the ER at the same time. And why is that a massive problem here? Yeah.
Because you're getting endogenous antigens being put in the ER for class 1.
Mm-hmm.
So why do they not bind to class 2?
Yeah. You basically shoot peptides into the ER. And this drawing draws, looky, MHC class 2 stays empty. and MHC class I doesn't. And in reality, sometimes in the textbook they draw a line down the middle of the ER. There's no line in your ER. Those peptides can diffuse perfectly well and come get on MHC class II. And so if things really worked exactly as was shown here, then MHC class II would get filled up with peptides in the ER, and it would make it here where it's supposed to bind antigen, and it would already be full. And so we couldn't pick up antigen because it was already full. And so the problem here is actually, how do we deliver the MHC to the peptide in a way that it's not already full of class I's peptides? And so that becomes a real issue here. So it's not just about how do we get the peptides to the MHC. That's actually really easy here. It's how the heck do we get a MHC that's ready to go to the peptide? And the way that we do that is that MHC class II is working with another protein. That other protein is known as the invariant chain. And so this is what MHC class II looks like when it is being biosynthesized in the ER. So MHC class II is biosynthesized with a partner protein called invariant chain, which is sometimes abbreviated as II. And invariant chain has a cytoplasmic tail, but also actually has a part that goes through and binds or covers, but it's really binding in the cleft. And so this is really kind of how MHC class I looks, or sorry, MHC class II. We've got invariant chain in the cleft. It actually pulls together 3 MHC class IIs, so we get this trimer. And there's a little piece that's out in the cytoplasm, and that little piece of invariant chain acts as an address label that says, take this to the endosome, and kind of bring this to the late endosome, the place where the peptides are coming. So part of the way we get the delivery is through invariant chain. Because invariant chain both blocks the cleft so that class I's peptides don't get in there, and provides an address label that says, send this MHC to the part of the secretory pathway where those peptides are coming. So invariant chain is pretty important. As you might guess, it does not vary. It is invariant chain. And so this is the actual picture from your textbook. So MHC is really biosynthesized in the ER along with invariant chain. So you can see invariant chain is plugging up the cleft so that these peptides can't get into class II. They can only get into class I. As class II goes through the biosynthetic secretory pathway, it gets degraded a bit. So you can see that it gets degraded and goes away until at the end, MHC class II is just left with a little piece of invariant chain covering the cleft. So we get rid of all the address label pieces, and we end up just with this little piece plugging up the cleft. That piece officially is known as the class II invariant peptide, otherwise known as CLIP. And so when class II gets to these peptides, it has CLIP in the binding cleft so that there aren't any other peptides that came from anywhere else in that binding cleft. The P in CLIP stands for peptide. I told you before that MHC class II plus peptide is a super stable molecule. SDS stable, in fact. So now we've solved the problem of keeping peptides that shouldn't be there out of class 2. But we've created a new problem. What's our new problem? What problem do we have now? Yeah, Sebastian. Now you actually have to get rid of clip. Now you get clip out of that groove. And this is a really stable set of 3 proteins together. And somehow you got to get rid of one, get one of them out of this complex, even though it's crazy stable. It's SDS stable. And so we have a mechanism to do that. Everyone always loves this mechanism. And to tell you about this mechanism, I need a volunteer. Jamie, would you like to be a volunteer? Jamie, come on up. Jamie already knows about this, so. She was briefed. All right. So Jamie is MHC Class I. And she is holding clip. So there is our MHC Class I molecule holding clip. She would like to be holding a peptide, but she cannot because she has clip, right? So what we have to do is we have to figure out a way to get the peptides there. So now Jamie is in the location where peptides are. And we're going to talk about the location in a bit. So we're in the right location. This is honestly what happens. There's this other protein. I am the other protein. And the other protein does this. Now drop it. That's what happens. I'm completely 100% telling the truth. There's another protein that basically bumps into class 2 until it drops clip to test affinity. Excellent job, Jamie.
Speaking with the dean.
Okay, there was a famous time in a past year about this. No, but my shoulder hurt for the rest of the day. The one time. I'm sorry I hit you hard. I think I did use Mark for the demo when Mark took the class. It was either Mark or Rob. They were both in there. So the name of the other protein that I was pretending to be, in the case of humans, is hladm. You know it's human because it has HLA. The name sounds like it's a class 2 protein. It's actually not a class 2 protein. It's just class 2 associated. It is encoded in that area of the genome, but it doesn't present peptides. It just helps class 2. In the case of mice, the protein's name is H2M. You know it's mouse because it has H2. Ends up being called H2M. And so what happens is hladm comes to the same compartment where MHC and the peptides are, and physically interacts with MHC, knocking the peptide off. And so you can also see that happening in this figure from the textbook. MHC class 2 is SAD. It has clip in its path, or in its cleft. DM interacts with it. That bumps the clip off and lets a peptide go back in. And in fact, this interaction continues until there is a peptide that is so stable in the class 2 cleft that it cannot be dislodged. And so this kind of helps us make sure we have a very stably bound peptide. And so the binding is due to the action of hladm, otherwise known as H2M, who has previously in this class been referred to as Marshawn Lynch. So the other question that people sometimes have is where exactly this happens. Is this in the lysosome? Is this in a late endosome? Like, what's the deal? And immunologists have done experiments for a really long time to figure out the answer to that question. And so people have been trying to figure out which of these many compartments is the compartment where this process happens. And in the end, what they have realized is that depending on how you do the experiments, you get different answers. And it's a really hard experiment to do. And in the end, we gave up, and we named the place where it happens the MHC Class II loading compartment. And so we just made up our own name for a thing. that MHC Class II loading happens in the MHC Class II loading compartment. If you look, however, at a lot of the data about the MHC Class II loading compartment, you will notice that it has a whole bunch of little vesicles inside of it. And it really, most data seems to indicate that this is a multivesicular body, which we don't know a ton about. And so maybe there's different kinds, and that's why we can't quite figure out what's up. But it seems like this is a multivesicular body where this process is happening. And so now you can kind of see all of these steps, except for our final step of MHC Class II. And the paper that I'm going to show you some data from was published while I was in grad school. I knew the person who is the first author on it, and every time, I still laugh to this day, and we're almost 20 years out on this paper. MHC Class II is not automatically or constitutively displayed. The cell actually has to be triggered to put MHC Class II on its surface. When the cell gets a signal that starts to trigger it to put Class II on its surface, that MHC Class II, which is in the M2C, the MHC Class II compartment, is actually pulled along microtubules to the cell surface. And so there is this active process where the cell will put MHC Class II on its surface. It's not happening all the time in these cells. And so here you can see this is kind of a cell that is at rest. The green is the Class II, and you can see the Class II is kind of all over the place. When that cell gets stimulated by a PRR, suddenly we put all the Class II on the surface. And so, in fact, this is another place where we see an action of PRRs. And so you can see that this is MHC Class II presentation is an active process. This figure is actually stills from a movie where they watched cells do this process. And so the green is our Class II molecules. Here you can see the cell starting to get a signal, and you can watch the microtubules actually extend down to touch the cell and be like, here is your Class II, you may see it. And then after the signaling is done, retract away. And so this is all done on microtubules. If you read this paper, Marianne Bose made up a name for this process. I can't even write it. The name for this process, according to Marianne Bose, is tubulation. That's not a word. I'm sorry, Marianne. So I will just say it's an active process using microtubules, where the cell uses its microtubules to put the Class II on the surface of the cell, though there are parts of the literature that refer to this as tubulation. This MHC Class II will present to T cells that have CD4 on their surface, because CD4 is needed to help bind and stabilize to MHC Class II. And so MHC Class II turns on CD4 positive T cells, and those CD4 positive T cells are the helper type of T cells, which produce cytokines to influence other leukocytes. So that's what's going on here. And so this is the lovely figure from the textbook that summarizes all of the steps of both the Class I and Class II presentation process. And so everything I've just been telling you about is summarized here. So the rest of the slides that I have are kind of about what I think of as the asterisks to this process, or the places where there are sort of exceptions to rules, or things that are sort of interesting about this process. I'm going to sort of skip around because I'm looking at the order of the next couple slides, and that's definitely not the next place I want to go. Who thought that was a good idea? All right, so the next place I'm actually going to go is here. This is where I ended last time with MHC Class I presentation, which is the fact that I told you that self-peptides are always being presented on Class I all the time. Way back at the beginning of this semester, I told you that sort of people are in some cases thinking about the immune system more as the system that assures homeostasis as opposed to like one about pathogens. And so this is actually a way the cell is saying, I am healthy. This is kind of a homeostasis mechanism you can think of. This is not quite as true on Class II because we have to actively be presenting on Class II, although when the Class II molecule is picking up peptides, they are sometimes self-peptides. There are some self-peptides that are in the lysosome just because the lysosome is a trash can. So there are self-peptides presented on Class II, but not as much as Class I, where we're presenting self-peptides 24/7, including right this second by all of your nucleated cells. See all of your nucleated cells. And this process is incredibly important to the cells. And this process is incredibly detrimental to viruses because those viruses get recognized, and then the cells that are infected with them get killed by CD8 positive T cells. As a result, viruses try to avoid this pathway in all sorts of different ways. And so these are some of the ways that viruses evade the MHC Class I presentation pathway. Many of the things I'm going to show you, in fact, all of the things I'm going to show you for the next few slides, and these are examples from this textbook, all of these examples, and all the examples I'm really going to show you except for one, are viruses that have in common that they're all super big viruses. They got lots of proteins, they got big genomes, they got room in their genome for all sorts of fancy things. I'm going to show you one that doesn't have fancy things later, but right now these are all viruses that got fancy things. So one example of a virus that can avoid the MHC Class I presentation pathway is EpsteinBarr Virus, which is the causative agent of mononucleosis. It can also cause some kinds of cancers. And Epstein-Barr has a protein known as EBNA1. EBNA1 is made in many, many, many copies by cells that are infected. So you could imagine there's a ton of it in cells. So it could make a ton of peptides to go onto MHC. That would be bad. And so in fact, EBNA1 is kind of interesting because it has these weird patches of glycine and alanine repeats. See, you can see the G's and the A's. And as a result, when EBNA1 goes into the proteasome gets gummed up. For some reason, the proteasome can't really degrade this protein very well. As a result, we don't make peptides from EBNA1. And so EBNA1 can never get presented on MHC Class I. The cell keeps on presenting self-peptides just fine, but it never presents EBNA1. And so the virus is basically hiding from CD8 T cells. And so in this case, we are seeing a virus that is evading the step of proteolysis. They can't really evade like infecting because that's kind of the point of their life. But pretty much as early as this process as we can think of, we can find evasion, like as early as possible by this proteolysis. Some of the more fun ones are these. These are actually two very famous proteins in immunology. One of them is called US6. It's from a virus called HCMV, which is human cytomegalovirus. Another one is called ICP47. It's made by herpes simplex virus. These two proteins work conceptually in the same way. They're very, very similar. And if you actually look at the structure, it makes me a little sad that they didn't draw them quite like this in the book, because this is really what they look like structurally. These proteins look like a stopper. And these proteins just make a stopper that plug tap. So we can no longer shoot peptides into the ER. And so MHC stays empty forever, sadly. And in the end, it never ends up on the surface of the cell, because tapicin doesn't release it until it's stable and ready to be presented. And so the MHC just gets sort of grounded in the ER with no peptides ever. And so these viruses evade MHC at the level of delivery. In fact, these two proteins and figuring out what the heck do these viruses do is how TAP was discovered. What you can also realize in this case is that if a cell is infected with one of these viruses, with one of these proteins being active, not only will we not present peptides from this virus, we will not present self-peptides either, because all of the TAP is blocked. So no peptides make it into the ER, whether they're self or viral. And we're basically stopping any MHC from going to the surface in the case of these cells. There's another virus, Adenovirus, that has a protein called E19. E19 actually gets in between class 1 and tapicin. So now class 1 is not in the right place to get any peptides. And it's not being held in the correct structure for binding. So this virus evades at this step. So basically, if you can think of a step, there's a virus that evades it. And you can also imagine that in this case, the peptides are not going to ever make it to the surface, nor is the MHC ever going to make it to the surface. So self-peptide presentation and viral peptide presentation is blocked here. Another example is that we have this virus, murine gamma herpes virus, that makes a protein called MK3. MK3 is kind of fun. MK3 binds to the MHC class 1 in the ER and adds ubiquitins to it until it gets pulled out of the ER and thrown in the proteasome. So this virus basically makes all the MHC in the cell get thrown in the trash. So it's a good way to escape getting yourself presented on MHC. You can imagine it also is going to stop self-peptide presentation. Similarly, there's actually another one. There are a couple other ones that actually take the MHC when it's trying to go to the cell surface and throw it in the lysosome. So they get rid of the display step. So every single step here is in fact inhibited pretty much by some virus. And so here you can see all the different steps being inhibited by some different viruses. In fact, this strategy is so common. So many viruses have evolved ways to do this that we actually have a whole other arm of the immune system that recognizes cells that don't have MHC class 1. And if you don't have MHC class 1, you're messed up, and we kill you. And so, in fact, it is incredibly important for homeostasis that self-peptides are always being presented. If any of these cells stop presenting class 1, then they are killed with the assumption that there's been sort of a viral infection. In fact, if a cell actually is a cancerous cell, it often loses class 1 too for other reasons. So it's either it's a tumor or it's infected. There are also some other ways that viruses can evade this process. So this is an example from SIV, basically like HIV in monkeys. This is a really tiny virus. It does not have room in its genome for any extra fun stuff like stopper proteins or other evil proteins like the ones we've just seen. Instead, the way that this virus escapes is that there is this epitope. The amino acid sequence is shown here. And particularly right now, we are looking at some macaques, some monkeys that have a particular MHC Class I type. This is the immunodominant peptide that is presented by the MHC Class I type. So this is the antigen that the T cells often are recognizing in this HIV case. And so you can see that, in fact, in this monkey, the monkey got infected, it had a bunch of virus, and then the virus went away. Hooray, the immune system was winning. And the monkey kind of had okay CD4 T cells because the immune system was winning. And then suddenly, the virus came back. The immune system stopped winning. And then the CD4 cells went away because the immune system stopped winning. Why did that happen? Why was the virus suddenly able to win? Well, the virus picked up a mutation. It actually had a 1 base pair change. That 1 base pair change made this T change to an I in the protein. And it turned out that T was the anchor residue for this peptide to bind to the MHC Class I. And so by the virus mutating, so that it just got rid of the anchor residue, its peptide couldn't be presented anymore. And so mutation and variation is the way that other viruses will evade MHC Class I. All right. I've got a few other kind of interesting exception things. So I'm going to go into a different one quickly right now. There are also some cases where we have evasion of MHC Class II presentation. Sometimes by messing with the phagosome or the lysosome, we mess with Class II presentation. A more famous example is the example where the microbe just punctures a hole in the phagosome and leaves, which kind of makes it hard for that to be presented on Class II. You can see that here, Listeria lysine is this protein that's made by this bacteria called Listeria. It makes a pore and allows Listeria to escape the phagosome. And this is actually Listeria's life cycle, like seriously. Listeria gets phagocytosed into a cell. Here you can see it got phagocytosed. Then it says, thank you very much, I don't want to be in a phagosome anymore. It pops holes in the phagosome and gets out into the cytoplasm. Here you can see it in the cytoplasm. Then it says, this has been a nice home. I like this very much. My progeny need a new home, thank you. And it actually polymerizes actin. Behind it, you can see the actin polymerization here to make what is known as a comet tail. And it just pushes its way into the next cell. It never has to be extracellular ever again. It just pushes from cell to cell, breaks out of all of those membranes, lives in the cytoplasm happily ever after, pushes into the next cell with the comet tail when it wants to go to the next cell, and on and on and on. Yes, Sydney?
Deal with that infection, because most antibiotics aren't metabolized by our cells.
Correct. You hope that, so antibiotics probably going to be a problem for this. There are some antibiotics that work for it, but it's a rare set of antibiotics. Class II presentation, not going to work for this. Looking at this slide, can you imagine something that would work for this? So there's some with some PRRs. There's one other thing. This is a great scheme if you want to avoid Class II. But Class II is not the only MHC in town. By breaking out of the cytoplasm, you don't get presented on Class II very well. But you get presented on Class I super well. And so Listeria is like this weird bacteria that gets ends up having a response that looks almost more like a response to a virus than a response to a bacteria because of this evasion. And so Listeria actually has this whole MHC Class I response instead of Class II. So there are other things I could tell you about, but I'm going to skip to one other piece of this that I want to get to. At some point, I will have to tell you about the things I'm skipping, but it will not be today. I told you previously that T cells only respond to proteins. And there is an asterisk on that. I told you that MHC only presents proteins, and that is true. However, we have found that there is a way that lipids can be presented. So first of all, remember that many of our phospholipids in particular have one of these hydrophobic head groups and these long, skinny hydrophobic tails. You can see that structure here. And it has been shown that there is this other protein that presents lipids. That protein is called CD1. It's the first one of them found, so CD1. There are different types of CD1, so you can see them listed here. I'm just going to call them CD1. And CD1 is able to present different lipids. If we actually look at the structure of CD1, the structure of this CD1 molecule is almost identical to the structure of MHC Class I. So CD1 actually has one big sort of alpha chain, and CD1 needs to bind to the partner beta-2 microglobulin, just like Class I does. So in fact, people were at one point somewhat confused when they made mice that didn't have beta-2M, because there were some responses that were missing, that they're like, why is beta-2M mess this up? And it's because beta-2M is actually part of two processes, presenting peptides on Class I and presenting lipids on CD1. But here you can see the sort of structure of CD1 and the structure of Class I. You see they look pretty similar. There's a little bit of difference up at the binding cleft, but otherwise they're pretty much identical structurally. And you can see that sort of general difference in the structure here. So the only difference is how the binding cleft is shaped in CD1. Instead of having just kind of like that hot dog bun region, there basically are 4 big long channels that allow the different hydrophobic chains to burrow, to sort of be pushed down in. I was going to use burrow, but that makes it sound like they were doing it actively and trying. They're not trying. Lipids don't try. And then the hydrophobic head group sits up at the top. And so we can have lipid binding in these pockets of the CD1 binding cleft. If we look at where CD1 acts in order to pick up its peptides, so the processing steps for CD1 look exactly like Class II. So structure Class I, processing Class II. So CD1 picks up lipids in different parts of the endocytic pathway to present those lipids. In humans, we know that there are actually multiple types of CD1. There's only one type in mice, and so we don't totally understand what's up with the different ones in humans. We know a lot more about just sort of CD1 in general from the mouse. We think that the different ones in humans vary based on things like which tissues make them, as well as things like which compartments they're in. So which compartment gives them their lipids. We think that's where the variation is. We think that's why there's more than one, but we don't totally know. If you recall, MHC Class I shows its peptides to a particular type of T cell. What type of T cell does Class I show peptides to? Yeah. CD8s. So CD8 positive T cell. That means it's a T cell that has the CD8 molecule on its surface. MHC Class II presents its peptides to a specific kind of T cell. What kind? CD4. It's a T cell that has CD4 on its surface. CD1 also presents to a specific kind of T cell. The T cells that recognize CD1 are called NK T cells. These usually confuse the heck out of people because they're like, well, I learned about NK cells, what's up? These are T cells that have some similarities to NK cells. They're T cells that have an NK cell marker on their surface. And honestly, the main definition of an NK T cell at this point is a T cell that recognizes CD1, because there's so many other things that are wacky about them. So lipid antigens presented on CD1 are recognized by NK T cells.